I'm going to start out with a brief introduction into salt cedar and the biocontrol project, which is probably common knowledge for most of y'all, but please bear with me. Salt cedar is an invasive species of tree native to Asia and the Mediterranean. It was brought to North America in the early 19th century to be used as erosion control along riverbanks. Is that better? Okay. It was brought to be used as erosion control along riverbanks, but it quickly spread to dominate over 2 million acres in the United States and Mexico. Salt cedars increase soil salinity, and they also increase evapotranspiration, and this causes inhabitable conditions for native species along the affected riverbanks. So what's biocontrol? Biocontrol is the use of one organism to impact the growth or dispersal of another. Salt cedar biocontrol began in 2001 with the release of the salt cedar leaf beetle in Nevada, Colorado, Utah, and Wyoming. It resulted in excellent control of salt cedar in over 200,000 acres. And following this success, the salt cedar leaf beetle was introduced to Texas in 2008, where it's been seeing positive effects along the Rio Grande. Once the beetles are released, they defoliate a significant portion of the salt cedar, and after several cycles of this defoliation, the trees begin to die back, and this allows native species to return to the community. The purpose of my study was to evaluate the impact that the biocontrol project is having on insect populations, and this is important to assess the functionality of the newly arising ecosystem. It was hypothesized that the control of salt cedar would increase overall biodiversity, and it was predicted that the controlled areas therefore the salt cedar is making up a smaller portion of the vegetative community, it would show greater insect diversity. And conversely, the areas that are still under management for salt cedar would still show signs of reduced biodiversity due to limited plant diversity in the area. This study was conducted at 4 biocontrol sites along the Rio Grande. This section of the Rio Grande is just northwest of the Big Bend region of Texas. We chose 2 sites to be considered as controlled salt cedar populations. This was sort of chosen by areas where salt cedar is less abundant in the area, and this was the Farr property and Rancho Pensado 2. And then we also chose 2 sites that were still considered to be under management, therefore salt cedar was making up a greater amount of the vegetative community, and it was also showing flowering foliage. And this is Alamito Creek and Rancho Pensado 1. Insect collection began in March and continued through July of 2015. We used sweep netting, and this was conducted in 45-minute sessions per site with a total of 8 trips. This is with the exception of Rancho Pensado 2, where there was a lack of collecting due to flooding in the area. We also used pitfall traps, and this involves placing a cup in the hole in the ground with the rim flush with the ground and adding water and a small amount of dish soap to reduce surface tension. We placed 5 traps at each site along natural insect pathways. The specimens, they were left out for 24 hours, and then the specimens were removed and the traps were removed. And this was conducted once per site, with the exception of Rancho Pensado 2 due to flooding in the area. We also did black lighting, and this was conducted by hanging a single sheet between 2 trees and attaching a black light. The black light was ran for 2 hours between 2200 and 2400 hours, and this was done once per site type. Once collected, the specimens were euthanized in ethyl acetate, and then they were frozen in plastic bags and vials until processing, and then the insects were pinned and identified to the lowest taxonomic level possible. There were 14 insect orders identified from our sampling, with 52 families being represented in our controlled areas and 47 families being represented in our under management areas. And interestingly, if we look at our coleopteras, which are your beetles, we see a high number of families represented in each group. But when we look at our hemipterans, which are your true bugs, and our Lepidoptera, which are your butterflies and moths, we see nearly twice as many families in the controlled area sample versus the under management sample. Here we can see the differences visually. In our controlled sites, there's a much more diverse representation of families, but when we look at our under management sites, there's much less diversity and many more repeated samples. Simpson's and Shannon-Weiner diversity indices were calculated for each site along with an evenness value and a Jacquard coefficient. The red boxes denote the highest values per calculation, but it is important to note that the values for Rancho Pensado 2 are skewed due to a lack of collecting. The boxes on the far right, these ones, are the combined calculations per site type. And here we can see that no matter the measure, the controlled areas have higher diversity versus the under management sites, and even our evenness value is higher, indicating a greater diversity of families. So again, we collected 52 families for our controlled area sample and 47 in our under management sample, and 31 of those families were in common. This means that there were 21 unique families in the controlled area versus only 16 in the under management. We can relate this to our Jacquard coefficient, which is a numerical representation of community similarity. A Jacquard coefficient is a number between 0 with no species in common, with 1 being all species in common. And we see here that our controlled area sample value is very low, indicating that there were few repeated families. And our under management area number is a little bit higher, indicating less diversity and more repeated families. Here we're just comparing our two diversity indices, and we see that they both show greater diversity in our controlled areas versus our under management areas. So a higher H value indicates higher diversity, and evenness is calculated using our H value. Evenness is a value between 0 and 1, with 1 being completely evenly distributed species. So as communities become more diverse, evenness should increase. We can see from our results that evenness and diversity are higher in our controlled area sample than in our managed areas. So that means that the controlled areas not only had a greater number of families represented, but the individuals in the community were more evenly distributed among those families. Overall, we found that species abundance, diversity, and evenness were greater in the controlled areas compared to the under management areas, and this supported our hypothesis and our predictions. And this is very encouraging for our local saltpeter management because it provides evidence that saltpeter biocontrol is increasing our local biodiversity, and future studies should see if this change in biodiversity can be seen in other taxa, such as mammals, reptiles, birds, and plants. And I will be continuing this research to include plant data in the following months. I'd like to recognize Dr. Christopher Ritchie, my mentor for this project, as well as the Sul State University McNair Undergraduate Research Program, who funded me, and to the Tamarix Coalition for allowing me to participate. Do you have any questions? I don't know if there's a microphone. How much dieback there was? There was a lot. There was a picture in here. I didn't really tell you that. You could see, I mean, as you drive along the river road, you can see where the river is because of all of the gray dead saltpeter. In our areas, we even, our beetle counts are way down because our saltpeter count is way down. So even like in the areas that were considered under management, the trees that were still there were like old growth saltpeter, like the ones that were big, and there weren't many new ones. Any other questions? Yeah. Well, okay, so in the managed sites, do we have more of the beetles? I honestly don't know, because in my collection, when we were designing this project, we decided to exclude beetles from it. So all of the data in there did not include the Diorhabda beetles. So, but when I was collecting, I didn't even collect any to not count. So I didn't even see any over this 5-month period, for none of the, if we look at the map again, way down the southern kind of side of our biocontrol area is where they've been seeing more. But and into like Big Bend Ranch State Park, they've been seeing some, but really our beetle counts are way down. Well, thank you.